Hello students, welcome to this session. Let's study cell, part 4, cell division. This of the cell cycle. So there are three states, resting, interphase and cell division. So in resting state, there is phase is gap 0. The abbreviation is G0 and it is a phase where the cell has left the cycle and stopped dividing. Interphase gap 1, synthesis and gap 2. So gap 1 is G1. Cell growth and G1 ensures that everything is ready for DNA synthesis. Synthesis is phase, that is DNA replication. Gap 2, G2, growth and preparation for mitosis. The G2 ensures that everything is ready to enter N, that is mitosis phase and divide. Then cell division, mitosis, N, cell division occurs. The metaphase ensures that the cell is ready to complete cell division. End result, mitosis, two cells having the same number of chromosomes as the parent. Meiosis, normally four cells, each with half the number of chromosomes as the parent. Function, mitosis, cellular reproduction, growth repair, asexual reproduction. Meiosis, production of gametes in sexually reproducing eukaryotes with diploid life cycle. Where does it happen? Mitosis, all proliferating cells in all eukaryotes. Meiosis, almost all eukaryotes, animals, plants, fungi, protists in gonads, before gametes in diploid life cycles, after zygotes in haploidic, before spores in haplodiploidic. States: mitosis, prophase, prometaphase, metaphase, anaphase, telophase. Meiosis: prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 2, metaphase 2, anaphase 2, telophase 2. Genetically same as parent. Mitosis is, meiosis no. Crossing over happens. Mitosis very rarely. Meiosis is, normally occurs between each pair of homologous chromosomes. Pairing of homologous chromosomes. Mitosis no. Meiosis is. Cytokinesis. Mitosis occurs in telophase. Meiosis occurs in telophase 1 and telophase 2. Centromere split. Mitosis occurs in anaphase. Meiosis does not occur in anaphase 1 but occurs in anaphase 2. In mitosis, the prophase chromosomes entangle and condense. Two chromatids attached to the centromere can be seen clearly. Mitotic apparatus constitutes spindle fibers and asters. Metaphase, complete disintegration of the nuclear envelope. Two sister chromatids attached by the centromere align at the equator, that is metaphase plate. Each chromatid is attached to spindle fibers from opposite poles at kinetochores. Anaphase, splitting of centromere and two sister chromatids separate and go toward the opposite poles. Sister chromatids now become daughter chromosomes. Telophase, chromosomes cluster at opposite poles and recondense. Nuclear envelope develops, and around each cluster of chromosomes and two daughter nuclei are formed. Cytokinesis, separation of cytoplasm takes place. So after two nuclei are formed, cell organelles get distributed between daughter cells. Meiosis 1, prophase 1, it is extended stage and further divided into various stages, that is leptotin, chromosomes start condensing, zygotin, pairing of homologous chromosomes, pachetin, crossing over takes place between non-sister chromatids, diplotin, homologous chromosomes separate from each other except at the crossovers forming chiasmata, that is X-shaped structure. Dikinesis, nucleolus disappears, chiasmata separate, and nuclear envelope also starts degenerating. Metaphase 1, bivalent chromosomes align at the equator and homologous chromosomes get attached to spindle from opposite poles. Anaphase 1, homologous chromosomes move to opposite poles. Unlike mitosis, histochromatids remain attached. Telophase 1, nucleolus and nuclear envelope reappears, chromosomes collect at the poles. This is followed by cytokinesis and haploid cells are formed. Meiosis 2, Meiosis 1 is followed by short-lived and transient interkinesis. There is no DNA replication at this stage. Meiosis 2 is more like mitosis, and each haploid cell forms two more haploid cells. So after both meiotic divisions, there are total four haploid cells. Prophase 2, the nuclear envelope disappears. Metaphase 2, chromosome align in the center and kinetochore of sister chromatids get attached to spindles from opposite poles. Anaphase 2, centromere splits and sister chromatids separate. They move to opposite poles. Telophase 2, nuclear membrane reappears, cytokinesis follows, and we get four haploid daughter cells, that is tetrad.
Hope you find this session useful for your understanding and for your exams. Thank you.